welcome to the 8th vodcast in the information domain. In this one, we are going to look specifically at what happens when the chromosome counts of cells are off after fertilization. So in essence, errors that exist during meiosis and what effects that has. So the big questions: how do we change genetic material? And how do changes in that material sort of lead to changes in the traits that the organism has? So of course this is really springboarding us into all of our discussions on inheritance. So how it happens. One of the main ways that chromosomal errors and abnormalities occur is through a process known as nondisjunction, as well as mega sort of rearrangements that occur with chromosomes. So first and foremost, nondisjunction is when chromatids don't separate properly during meiosis. And so, for example, in this one, you can see that one of the homologous pairs in the first division does not separate properly. So instead of separating the pair, it goes right in to that cell, and then you get the further divisions. Okay, so it can happen during meiosis 1. You can also get nondisjunction during meiosis 2 when the two sister chromatids don't separate. And this leaves one of the cells as a haploid cell, but +1, because instead of having just one of each chromosome, it actually has 2 of one of them. Not necessarily a homologous pair of them, it just has 2. And then leaves a separate one of the other gametes in that process less than one than it's supposed to have. So if this were a human, you would get in some gametes, you would end up with 47 chromosomes, you would end up with 24 chromosomes, okay, and in some, you might end up with 22. Okay, so this is what happens. It's a lack of separation during anaphase, either anaphase 1 or anaphase 2. And you get that irregular chromosome count. And this has to happen in the germline cell to cause any problem. I mean, think for a second why that needs to happen. Because, I mean, why would that need to happen? Well, because that's when you're making those haploid cells, and if it happened in any other, there wouldn't be any effect from there. You also get the large-scale rearrangement, where segments of chromosomes also get altered during DNA replication. We've talked of this, like mega sections of the chromosome get deleted or rearranged. And so there's a couple of options. You can get a whole section being deleted, so it just chunks out a chromosomal segment, and that could include multiple numbers of genes. Or a couple of segments could be duplicated. That can happen. You can also get inversion, where certain segments, like it says BCD, get rearranged, and all of a sudden now you have DCB. That can happen. This causes shifts in alignment of chromosomes. Then there's translocation, and this one is where you get areas of one gene jumping. Okay, this is what we call, when we've talked about jumping genes before, this is what happens with jumping genes. Area of one chromosome jumps to an area of a non-homologous chromosome partner. Okay, so the key big question is, what are these changes going to do to the organism that's produced, right? So that's kind of always the big question. So what's the effect? You know, because you might say, well, okay, so who cares? So what do we get from that? Well, when it's a single chromosome, we have, a single chromosome change, either one more or one less, we call it a some. So you can either, because of one of your pairs, you're going to have a trisomy, or you might have a monosomy, and this involves with extra or missing chromosomes. Oftentimes, you're going to get a miscarriage, and this is sort of, I guess, it's nature's way of making sure that an incorrect chromosome count doesn't work its way through the generation line. However, some of these do result in creating an organism. So one of the most sort of familiar trisomies in somies is trisomy 21, which is also known as Down syndrome. And in this case, we have 3 copies of chromosome number 21. They result in a number of symptoms that arise from that, anything from a characteristic sort of facial appearance. There's varying levels of mental acuity and processing that goes on in it, and you get a plethora of other effects. There can be anything from heart troubles that can go with it to there's a lot of effects from just having this 3rd dose. I will say, from the individuals that I have known who have Down syndrome, I will say that getting a triple dose, and this is my experience, a triple dose of chromosome number 21 also gives you a triple dose of being some of the most compassionate, caring individuals that I have ever met. And So I don't know if that, maybe that's where your love gene is, but yeah. So along with these other things, I would say getting 3 chromosome number 21s makes you the most loving and compassionate person ever. So in this case, it is tolerated for an organism to be created. And then you get this question as why? Why for this chromosome number? Why can we manage creating a full organism who can have a fairly significant lifespan from this? And that's something that's been asked quite a bit. We also have sex chromosome somies, because like chromosome 21, that's an autosomal one. Here we can have ones where you can only have one X, okay, and that's known as Turner syndrome, monosomy X. So a girl would only have one X chromosome. Then there's Klinefelters syndrome, where you have XX, multiple X's, and a Y. Generally, all during due to non-disjunction events that occur. So you have here, and I've actually taught a couple of individuals with Turner syndrome over my career. You really wouldn't know it to look at them. They are phenotypically female, and that's because there is no no Y chromosome at all. They do tend to be sterile, and that is the case of a lot of chromosomal abnormalities. And that would have to do with the inability to make viable gametes. And there are some associated heart defects that go with it in some cases. And then there's the Klinefelters. Okay, and in Klinefelters, you get a phenotypic male, and that's because the Y is still present, but you get multiple X's, anywhere from XXY to XXXXY. It's been huge. You can have, because of the double dose of the X, form some secondary female characteristics just due to, I think, the production of more estrogen. But, that's, kind of rare in that case. And there are issues where you have multiple Y, multiple X, okay? Multiple Y, Y is your teeny tiny chromosome. We've seen this, very few genes, very early direct development, very, very early on, okay? So it's kind of important for that case. And so there is not so much an issue when you have multiple Ys. Multiple X's, though, do cause a little bit more problems as a result of that. So here's multiple Y that I just mentioned. Multiple X, huge chromosome, lots of genes. Both males and females have to function. Males only get a copy of it. We know that. And then there are certain things that on the X chromosome, you can have some areas of inactivation and activation of the chromosomes that end up coming into calling bar bodies. And this is why calico cats are generally female. It's an X-linked piece, and the only way you get that calico color is due to having 2 X's, okay? And having 2 X's, one is activating certain areas of pigmented color, and the other ones are inactive of black color. So you get this speckly, orangey, black, white color that exists in calicos, okay? Now, I will say this. They're not always female. There have been rare instances of calico males, and that's because when we looked at, and when a calico male exists, it is because they have the sort of cat version of Klinefelters. They have double X, and they have their Y chromosome, but they show the calico color. Kind of cool. So there is what's going on with a calico kitty. They're so cute. So we can also have whole sets of issues in terms of chromosomal, and this is in terms of not being a somy, which is a single body. We have a ploidy. And these mutations involve getting extra sets of homologous pairs. We as humans don't generally tolerate this. This is usually kind of a factor in with hybridization and things like that over time. We like to be diploid because we have 2 contributions from parent, one from each parent. We create gametes that are haploid. Okay, cool. We have that. We get that. But there's also organisms that are going to have a 3rd set of those, and they're triploid, and then you have tetraploid, all the way up to strawberries, which are octoploid and have 8 copies of every chromosome. So plants are far more tolerant of that change. And with rearrangements, sometimes we're not even sure exactly what happened. When you get shifts in actual rearrangements, like chunks of chromosomes going to another, sometimes you're not even sure what can happen. And because of this, they can often be silent things that happen, or some of them can cause some mega problems. So sometimes a translocation in a blood stem cell could lead to leukemia, okay? Because a section of normal chromosome 22 switches with a segment of chromosome 9, that causes an issue and you end up with leukemia, okay? So, and we call that the Philadelphia chromosome, okay? And then there's some things that you can look when translocations occurs, the diseases that can exist, and they're completely random. So we can't really predict translocation. Okay, so there you have it, gang. So from this, make sure you can do these couple of things. Explain how the mutations, explain the mutations, how they present themselves and arise, and explain the causes and effects of those situations. And get ready to start talking genes and talking alleles and talking heterozygous, homozygous, and all of that stuff. So take it easy. Have a great night, guys, and we'll catch you later.